from Microbe TV. This is Office Hours for January 1st, 2025. I'm Vincent Racaniello, and welcome to your viral corner of the internet. This is an experiment. I haven't done an Office Hours since December 4th for various reasons. One of them is Christmas, 25th. And then other reasons, but I wanted to do it, and also to start 2025 in a good way. But I also wanted to see if anyone would come, if people really loved their New Year's Day and have other stuff to do, or would they come to a live stream? And we have 64 people here, so thanks for coming. Thanks to our moderators who are here tonight. I saw Vanity Nutrition, Barb Mack. I saw Les and Steph San Francisco, Steph SF. Thank you all for joining us and making it civilized. Tom just showed up. And yes, tomorrow, January 2nd, is my birthday. So I get all this big stuff out of the way in the first 2 days of the new year. We get New Year's out of the way, right? And my birthday, and that's it. For the rest of the year, I don't have to worry about my birthday. And I'm going to be 72 tomorrow. I was born in 1953. And damned if I don't look 72. I don't think I do. You look the way you feel. So I feel much younger. And happy to do this tonight to get the year going, because it's symbolic, right? We have a whole year ahead of us of virology. Who knows what's going to happen? We'll talk a little bit about that. Anyway, tell us where you're from so I can say hello to you. January 2nd was Isaac Asimov's birthday too. That's right. I remember that. A few other people of note, right? So my mother went to the hospital on New Year's Day. And I was born at 6 a.m.-ish, roughly. I was born in Paterson, New Jersey, in St. Joseph's Hospital, which is where my father worked. Although, he was probably still an intern at the time. I don't remember. Maybe not. He may have gone off to do his residency in the Bronx. I don't remember. But not a big fan of holidays, not a big fan of birthdays. People can do what they want, right? But I prefer just to go along. That's why I don't mind doing a live stream on New Year's Day. We have a lot to talk about. Let's see. Who's here? Oh, Kip is here. He's in San Francisco. Kip and Laura visited the incubator last week. It was this past week. It was Monday, or Tuesday. And, it's always fun to have you folks visit the incubator. We always have a good chat, right, Kip? And thank you for coming. And we'll see you when you come back. Mark Martin is here. Ooh, I should turn on my phage. It's on this side. Turn it on. Yeah, it's always a cool thing. Oh, I have the remote, right? So let's see. There we go. Number 5. Disco phage. It's funny that it's a disco phage. We did a paper on TWIV this past week called Phage Discovery. Abdulaziz is in KSA, Kingdom of Saudi Arabia, 4C. A bit chilly, eh? I don't know what it is here. It got warmer these days. Yeah, it's currently 4C. Also right here. How about that? Wishing everyone a better 2025. I think 2025 is going to be challenging with respect to science and public health, as you know. But we are here to help you navigate it all, explain things, and tell you the scientific truth as we know it. No BS. no political usage of science or any of that stuff. We are here to do that. Frank wishes me happy birthday. Many happy birthdays ahead. I'd be happy to get into the 80s, right? Tony Fauci's in his 80s, right? Still going strong. I think it's possible. We will see. Oh, and of course, Happy New Year to everyone as well. Kang is in Chicago. It's -3 and cloudy, pretty cold there. So we have from Vanity, Acapella Sciences, Science Parodies Christmas Carols. There's a link. That's cool. Very cool. Very cool. Tell us where you're from. Kip, thank you very much for your support of science communication. Really appreciate it. Hello, John from Minneapolis, -7C. That's pretty cold. We had -12 last week here in the Northeast. Thank you all who are wishing me happy birthday. I appreciate it. Tomorrow, the 2nd, I will be at the incubator. Someone told me, you are nothing without the incubator, and the incubator is nothing without you. I think it's a bit of an exaggeration, but I would agree that I'm nothing without the incubator. I love going there and working. Lise is in Columbus, Ohio. Les, of course, says I'm turning 39 again. Actually, I don't want to be 39. Because I know a lot more now. I wish I had the body of a 39-year-old, right? Incredible health. But I think I'm smarter than I was at 39. Will is in Changsha, where it's 4 degrees Celsius. Cheryl, yes, you got it right. I'm going to be 72. 72. And I think in French it would be 72. But I'm better at the Italian. 72. Yeah, still a spring chicken. I try. Privyet, Veronika. 1 degree in Moscow. You have a lot of snow out there. Welcome back. I'm not elderly. Yeah, that was 75 and up at the elderly, right? It's an interesting question. What I should be doing at my age? I take respiratory infections seriously. So if I got COVID, I would take Paxlovid. Ooh, that's a little rhyme, isn't it? A couple of weeks ago, I had a cold, an upper tract cold, and I just took a COVID test, and it was negative. So, I mean, I waited too long. It wasn't flu. I could tell it wasn't flu. So it wasn't COVID. So I didn't do anything. Just took care of myself. I do try and mask in public areas all the time. I don't want to get it. I don't want to get sick. Yuhan Lee is in Shanghai, China. Welcome and thank you for the birthday wishes. Appreciate it. Sharon is in Tasmania. Pleasant summer, 13C. Just celebrated my 64th birthday, New Year's Eve. Well, that's great. Yeah, we should. I hope everyone has a kind and gentle year, even the animals. Even the animals. Yeah, 50 is a good time to be born. We got the Salk vaccine. So, yeah, I would have gotten it in 55, April something, 1955. The clinical trial results were released, and I probably got it shortly afterwards. But then I got Sabin in the 60s when they replaced the Salk vaccine. So I have both. Philip is in Wales, UK. Happy New Year. Dagfinn is in Oslo, Norway. Happy New Year. Tom is in Oregon, I presume. Nice intimate group for this evening. It's 120, 130. It's almost usual. Usually we're bumping up against 200, so it's okay. Yeah, it's a good number. We're going to have a good chat. I have a little mini lecture for you to kick off the year. And I'd like you to answer some questions for me. Think about it. Think about, what are you worried about this year in terms of viruses? Think about it. We can talk about it later. Yeah, John had a great promo video for Micro TV. We had several people sending in promo videos. I love them. I haven't put them all up yet. I have to get the rest of them up in the next week or so. We're going to have 2 more weeks of fundraising, and then we'll stop bothering you like PBS bothers you, right? Carol, our favorite nephrologist from California, 56, substantially warmer. That's cool. Got your Christmas card at the incubator with your family. It's lovely. Lovely. David is in Dayton, Ohio. Chris Lipowski, you're welcome. Look, I'll be in Montreal next summer. Or maybe we can see each other. Yeah, I have to tell you this. I haven't worn this. This is a very old sweater. The label says B. Altman. Does anyone remember what B. Altman was? Yeah, it was a department store in New York City. I don't know if it was elsewhere, but it was certainly in New York. I don't think it exists anymore, right? And it's a little snug, but yeah, it's red. So I thought, I wore it once last week for a clinical update. Thought it would be, actually I wear it because I wear a sweater. It's chilly down here in the basement. Arland Theodori, wow, welcome. I don't know where you're from. I haven't seen your name before. Thank you for the New Year's wishes. Where are you from? Yeah. Of course we're here. Yeah, you can imagine that you might not be here, and I would totally understand it. But as I said, I didn't want to miss another one. And so the experiment, and you're here. Patricia had her gallbladder out. I'm sorry. I hope you're okay. Good luck. Actually, I usually wear black, right? So I have a black shirt underneath it. But certain occasions, red is good. Red is good. Angela is from upstate Manhattan. Welcome. Logotic is from Chicago. And Martin is in Tacoma, Washington, where it's 19 degrees. You see his phage lighting away there? Yeah. I mean, it's blurry because it's out of focus, but it's the one he gave me a couple of weeks ago. He sent me a nice movie of a version of his. Yeah, it's a good one. I'll use it, Mark. Don't worry. I have good ways of using things. Leo is in Mendocino, California. Questella is with her kid, their dad, and I, all present. Well, welcome the Questella family. Questella, I have to tell you, Questella family. Questella was in the first live stream ever, which was quite a few years ago. And she said, don't worry, this will work. She had absolute faith. It's been here ever since. Thank you, Questella, for your faith. MN is outside D.C. Lolo's Lounge, thank you for the birthday wishes. Logotic is in Chicago. Yeah. Who else was born on January 2nd besides me and Asimov? Does anyone know anyone else? You can look it up. Wikipedia has all that. People born on January 2nd, right? And Angela, from another 72-year-old. Thank you very much. Come visit the incubator. You're in Manhattan. You could just get on the A, get off at 34th Street, and you walk 2 blocks, and you're at the incubator. Come by. That would be cool. Questella, thank you for your support of science communication. I really, really appreciate it. Hello, Jeff. Thanks for your wishes. Cesar is in Peru. Welcome. Carrie is in Michigan. Heather is in Kamloops, BC, Canada. Far west. You don't look 72. Thank you. Those are the best words you can say to me, right? But only if you mean it. If you're prevaricating, I don't want you to do that. Just be honest, okay? Really appreciate it. Truman 17 is in Fort Worth, Texas. A bit chilly there. A bit chilly. Patricia's in Massachusetts. Yeah, in Russia, it's already January 2nd for many hours. Yeah. I don't know. That's weird, isn't it? Like some parts of the world are in the future. It's weird. Yeah, it's January 2 in China. All that part of the world, right? China and all the countries above and below and left to the left of it. Russia, I guess all the way up to Eastern Europe. I don't know how far into Europe. Well, let's see, 6 hours, right? So some European countries wouldn't be tomorrow yet. Betty K. still here in Washington Heights. Here's Laura, who also visited the incubator last year. Thank you very much. Yeah, I hope 2025 brings joy, peace, and luck for everybody. I will be here trying to usher you through it. Jeff is in Elgin, Illinois, just getting over the flu. Should have gotten the flu shot. Well, you're getting over it. That's good. Next time you should get the flu shot. Yeah, it will keep you out of the hospital. You don't even look close to your age. Thank you very much. Thank you very much. Well, you know, yeah, some people my age don't look very good. I don't know what the key is. I try and lead a healthy life. I try to use my brain a lot. I try to exercise, although lately I haven't been. I try to eat properly. So maybe that helps. It also helps not having gray hair, right? I keep my beard short because it is gray. But if it's short, it's less noticeable. But the shock of brown hair, I do not color my hair, folks. I told you that before. I don't color my hair. I try and keep it short, but sometimes it gets long. But it is not gray. Sometimes there are a few transparent hairs, but it's not gray. And that's good. Oh, you think it's Italian genetics? Really? I don't know. I've known some 70-something Italians. They don't look so good. Well, bent over and emaciated and so forth. But thank you very much. Thank you. Rad is from Toronto. Rip de Camp is from Taos. The Isle of Man, it's already my birthday. Okay. Yeah, I guess so. 6 hours. So all of Europe, it's my birthday. Hans is in Guayaquil, Ecuador, 30C. A little hot. Thank you for your best wishes. Eliza is from Southern Ontario. Yeah, Podfather. Martin likes to call me the Podfather. Although there was a guy, Adam Carolla, I think, who really got podcasting popular. They call him the Podfather. But I'll take it. I don't mind. Yeah. This is the first office hours of the year. That's pretty cool. Leo 2MD is from Philly. Mike and Lori are in Seattle. Patricia saw a link to a Blogging Heads interview with a young scientist who argued the lab leak theory. Yes, this is nonsense. I get letters all the time saying, but you're wrong. You're not looking at the data. No, I'm not wrong. I am not wrong. I'm sorry. This is one thing that I know. I know viruses. I mean, there are not a lot of people that have been around viruses for as many years as I have. I don't know everything. But you look at the data, there's zero evidence for a lab leak. Okay, folks? Zero. If you think there is, you just don't know what you're talking about. End of story. And the whole congressional report, well, I'm going to read it tomorrow to see if there's nothing in there. I'm quite sure there's nothing in there. Because there is no evidence. All the evidence points to a natural origin. And there's considerable evidence, right? And you saw recently, Zhongli Shi sequenced all the viral samples she had in her lab. None of them were any closer to SARS-CoV-2 than RATG13, 1,200 bases different. That's what she said. So we don't have SARS-CoV-2, and they clearly don't. So come up with some other excuses why you think it might be a lab leak. Just make up stuff, without any evidence. I don't get why you want to do that. Why don't you look at the data that exist and go by that. Okay, enough of that. Maureen is in northeastern Ohio. We have so many people from Ohio. That's cool. That's cool. FSM is the dog is in Wales. Good revolution around the sun. That's a good way to say a year, right? Tom, University of Utah, Salt Lake City, back from North Carolina. What do scientists think sweating sickness was? Lots of theories. We actually talked about it on TWIV years ago. Nobody knows because there are no samples. It could have been a mix of things, right? Who knows if it's a hantavirus? I don't see any evidence for that. Bernard is in Salisbury, Maryland, starting to get chilly. Oh, I don't want to give anyone air who has a lab leak theory. It just gives them publicity. Patricia, there's nothing to it. And if I have that on, all they're going to argue is that I'm wrong on every point. And that's stupid because I'm not wrong on every point. And I had to look at the data and say, yeah, this is very strong data in favor of a natural origin. Yeah. Spamlet's in Bedfordshire, England, where the first day of 25 was about as gray and miserable as it can get. Oh, Abdul Rahman is from Brooklyn, New York. Welcome. Margot is in East Phoenix. Pretty warm there. Yeah, I'm full of science. If you want to hear science, come here. And other stuff, too. No, my birthday's tomorrow, January 2nd. This is Juni Shengri Kwai Li. Happy birthday. Thank you very much. Santa Barbara, California. Thank you very much. Mark is coming to the incubator in a month or two. I will stay out of political questions like best pizza, best bagel, or best pastrami. Well, we can find out what people think, right? Everybody has an opinion. But you do have to visit the incubator, Mark. I'm probably here March. I'm going to a meeting in Germany. I can't remember what it is, but it's just a few days. We'll see. We'll see. Thank you, Rob, for your support of science communication. The incubator rules. Yes, indeed. Spartacus is in Dorset, UK. Thank you very much. Lee says, Microbe TV, people have been asking me where I got my red DNA cable knit hat, which is my way to introduce them to Microbe TV. They're very lovely hats. I think they have a few left as well. If you have given 100 and you'd like a hat, please let me know. Lori is in Pueblo, Colorado, 31F. Nevitz Forever is in Eastern PA recovering from COVID and took Paxlovid. Mark says I should make a disco phage to donate for fundraising. Wow, that would be cool. We could do that. Yeah, I think there's a great community that's come together. I appreciate you mentioning that. And I think everyone likes it here, and I do too. I appreciate being able to reach out. Rach has been ill all over Christmas and New Year. I'm sorry about that. She was going to do a video. A bit too snotty at the moment. That's great. B. Altman, my mom's favorite department store on Fifth Avenue in New York City. That's right. Yep. This is old. It's ancient. Patricia is in Wisconsin, where it's 23F and -5C. I have some questions here I'm skipping over. I'll come back to them. Just want to see where everyone's from first. Microbe TV is on Blue Sky. So I haven't made a personal account. I have a Microbe TV account, but I have not been posting on it. I have a social media person. I only have 100 followers. But I think if I went on, I would have more. So I should probably make a personal account, or I can be snide. Right. Sue is in Southern California. Wayne is another 72-year-old. Yeah. Welcome to the club. January 2nd in Norway, also in all of Western Europe. Yep. MN saw a snippet of the Black and Yellow song. Yeah, that's what I say to people when they don't know how to pronounce my name. Just Black and Yellow. And they get it immediately. More than any repetitive pronunciation of Racaniello, I do. Black and Yellow does it. It's really amazing. And that came one year from one of my Columbia Virology classes ages ago. Students came up to me and, Professor, whenever we hear that song, we think of you. Black and Yellow. I said, well, you don't think of me, but that's nice that it sounds like it. That's cool. Very cool. Patricia's in Virginia. Carol wants to know, when's the best time for a Tdap for you? Child is due in August. You have to ask Daniel. Okay, Carol? Send Daniel at microbe.tv. He's the guy to ask. The ID doc. Will Red, Robert Reichs, The Common Good. Vincent epitomizes the common good. Blessings. Maybe I should read it to see what it's all about. Yep. What I learned today is from India. Red Hen 689 is in southeast Pennsylvania. All right, so Patricia's giving the advice to Carol. Thank you very much. It's 10 degrees C in India. Tess is in Canton, Massachusetts, 40 F and raining. Sandra says, I think you smile a lot and you have a great attitude, and this is why you don't look 72. Happy birthday. I think that helps. Yeah. I think smiling helps. I think having a great attitude helps, but I think also not having gray hair helps too. It's just my opinion. I don't know if it's right. All right. Let me just get through the welcomes. Neil Ricci is interesting. Backwards name there. From northeastern Ohio. New York Cohen is in Westchester. By the way, tell me, anybody in New York? No, there's this. I was walking down 7th Avenue yesterday, and there was this some kind of pastry shop called Crumble, and the line was around the block. I'm like, well, what the heck is this? Does anyone know what's going on there? G is in southeast Michigan tonight. Thank you. John, thank you for your support of science communication. Is Zelly an option? I don't think Jelly charges anything. You know what? Actually, our business account is not going to have any fees for anything. Like we don't pay wire fees because if we have a nice amount of money in a couple of CDs, so I think Zelly is good. And but let me check tomorrow, okay? Yeah, regarding COVID origins, yes, the U.S. intelligence agencies have no evidence. There's been no evidence that anything goes on. There's all this hearsay that's going on, but that's not science. Science is not hearsay. Yes. Yeah, 117. I know that Twitter is not an option anymore, but I do have 180,000 followers on Twitter, so it's hard to build that up quickly, right? Oh, yes. YouTube does it wrecked in yellow. Yep. Oh, you're welcome, Carol. My pleasure. Thank you for your contribution. We really appreciate your support. Maybe donuts. What about donuts? I don't eat donuts. Giles is in Portland, Oregon, where it's 47 F and rating. Crumble is expensive, fancy cookies. So 7th Avenue and like 22nd. The line was out the door, and around the block and halfway down the block. I mean, I would not stand for that. No cookie is worth that. But that's okay. Eleanor is also a 53er from New Jersey. Yes, in Austin, Texas, visiting your new grandbaby. Yeah, it's warmer in Austin, Texas. Okay, there were some questions here. Let me go back and see if I can find them as quickly as I can. Crumble is a big deal in New York City. See what I know? I don't know anything. I walk by and I see people standing in line, blocking the sidewalk, eating donuts or cookies. I mean, there was one lady, she had her box on a garbage can and she was just eating the cookies out of the box. Really? Really? So there used to be, and there still is, a Carlos's Bakery chain, and there was a store in Hoboken. The same thing, you couldn't get in. You'd have to wait an hour to get in. All right, let me find the questions. I have to go down a bit. So, yeah, a lot of people are worried about H5N1. So look, this is the beginning of January. In 2000, beginning of January, this virus was already spreading throughout China. So stuff could happen this year, right? I think, first of all, the pandemics are pretty rare, right? You have to admit they're not every year, maybe 10, 20 years. So the last pandemic we had before COVID was 2009 H1N1. Of course, AIDS pandemic ongoing. So, 20 years. I don't know that should say for sure what's going to happen, right? You could have one this year. I don't know what it might be. But many people are concerned about it. And here's this. Let's get to this first question here. Happy New Year. Happy New Year. I should have marked the questions. I should have starred them. Well, here's a Kang question. Come on. Here we go. If classifying flu viruses are the types using the letters A to D related to the enumerated H and N types, how are these types or subtypes identified? So only A has H and N subtypes, H1N1, H2N2. The B, the C, and the D do not have subtypes. They are classified, well, now they're classified separately based on the genome sequence. And the B is a separate set of viruses from A and from C and D. Originally, they were classified by antigenic cross-reactivity, but that's no longer done. It's done by nucleotide sequence. But no, H and N only has to do with the A influenza viruses. Okay, Patricia, do you think bird flu will mutate to become another pandemic? Well, I don't know, and there is no one on the planet who knows whether this is going to happen. If someone tells you it's going to mutate this year, they're full of it. Now, if it happens to, it's not because they knew it would. They got lucky, because you can't tell if it's going to mutate to become something that transmits very effectively among people. And that's what has not happened yet. We have known highly pathogenic H5N1 since the over 50 years. Emerged originally in birds and then first outbreak in China in people in 1997. There have been slightly over 1,000 human infections. And of course, in all that time, it has not mutated to become transmissible in humans. Now, it's infecting many more animals now than it was back then, right? The whole ecology has changed. It's no longer seasonal. The viruses are present in birds globally all year round. In the U.S., it's entered cows. It's infecting many other animals, and you probably saw the outbreak in the was it Washington Sanctuary of big cats? Half their cats, 20 or so of their cats died. Very sad. They're wonderful animals. So the more the virus reproduces in other hosts, the more it mutates, right? So many people say, it's got to mutate to a pandemic this year because it's in so many other animals. But no, not necessarily. It could very well be that the changes needed to transmit effectively in people are not compatible with fitness. Who knows? Nobody knows this. And again, if someone says they know, they're wrong. They're just BSing you. I don't think anyone can predict. However, we could be ready. We know that the flu antivirals work, both the neuraminidase inhibitor and the endonuclease inhibitor. So Tamiflu and zanamivir. Also, and the other one, whose name I always forget because I don't really use it too much, is an endonuclease inhibitor. They all work against H5N1, and we have vaccines that work. So it's not like we have nothing as we did when SARS-CoV-2 emerged. We had absolutely nothing. No antivirals, no vaccines. And it took quite a while. It took a year to get a vaccine, which was pretty quick. And in this case, we should have a vaccine in less than a year. So I think what we need to do is continue really good surveillance globally and have enough of these vaccines and antivirals ready. Not just for us, but for other countries as well. The world needs to get together and make sure that we're ready. So Leo 2MD, his worries or her worries for this year: breakdown of public trust for health policy from government academic institutions, and resurgence of disease from decreased vaccination rate. So I am also concerned about this. And many other people are concerned about this as well, right? I don't know what the situation will be with the confirmations. All of these individuals who have been nominated for various health and science and public health posts, they're all flawed, in my opinion. They're unqualified and they have features that Bhattacharya, someone asked about him. I've published a few videos on YouTube about all of them, except the Surgeon General, why I have problems with them. And Bhattacharya was co-author of the Great Barrington Declaration, where early in the pandemic, they said, you have to wear face masks. You don't have to do this shutdown and isolation. But more importantly, he believes that SARS-CoV-2 came from a lab. And I cannot have an NIH director who believes that in the face of evidence which says otherwise. I don't care what your political leanings are. The science says it came from nature. End of story. I'm not debating anyone on it because they're wrong. The whole House congressional report is wrong. It's just BS. So I'm worried about these nominees. Even if RFK Jr. doesn't get in, some other bozo will be nominated. It's less toxic and more acceptable to the, the Senate Republicans. I hope that there is enough of an institutional establishment that can counter bozos. So if a bozo said we should cancel the polio vaccine, there's enough smart people around to say, no, we're not going to cancel this because it works and we need it, right? I'm hoping that will prevail to some extent. But if it doesn't, then yes, we're going to have increases in infectious diseases. No question about it. Look, we've already seen measles cases. We had hundreds of measles cases in 2024. Shouldn't have any. Should be zero. It's all from unvaccinated kids. Parents who think it's a good thing not to vaccinate their kids against measles. All right, you want to have a choice. Fine. If you make a choice and your kid dies, that's on you because measles can kill kids. So it'll be followed by outbreaks of influenza, polio, hepatitis B. We will have massive outbreaks in this country, not in the first year, but eventually, if vaccination rates drop, because many of these people are anti-vaccine, decidedly anti-vaccine. They want you to buy their products, right? Even the Surgeon General nominee, the lady whose name I forget, She sells some homemade concoction. It's ridiculous. There's no evidence that it does any good. So I agree with you, Leo 2MD. It's going to be a tough year. Tougher than we all think, and not just in science and politics as well, but we're not going to go there. Yeah. Big bird flu affecting cows in Tulare County. What to expect with bird flu? So the good news is that all of this virus, H5N1 virus, circulating in cows, when it does infect people, the infections are rather mild. Now, that could change. But the idea would be, if you have so much virus replication in a new host, the cow, that leads the way to more mutation, and perhaps a virus emerges that can transmit really well in people. That's what you need. Right now, it doesn't transmit well. What you need are changes that allow the virus to transmit well, especially from asymptomatic people and during the incubation period, right? So, as I said earlier, we have no idea if that's going to happen or not. We have no idea. Nobody has any idea. And a lot of people like to say they know because they get attention on social media and they get invited to interviews. But they don't know. They have no clue. So I think the best thing we can do is be prepared. Let's see. What else do we have here? The common good. I have to remember that. If suddenly both innate and acquired immune systems are turned off, how long do you reckon a person could stay alive? Well, depends. You could put them in a bubble, right? And they could live because they would have no contact with anyone and they wouldn't get any microbes. Now, there are mice in the laboratory that have innate and adaptive deficiencies, and you can raise them under quasi-sterile conditions, and they're okay. Because if you let them out in the world, they would die. They would get infections for sure. So if you sent someone out, so if people had no, if they have no innate and acquired immune systems, they're going to die pretty early in age. But if you could somehow turn off the systems in someone and send them out in the world, I'd say within a couple of weeks, they'd be acquiring infections, which would completely debilitate them. The interferon system is really responsible for taking care of most infections. We don't even know about it because we get infected on a daily basis, and interferon takes care of it. And when you have deficiencies in interferon, and there are a number that are known, including making antibodies against your own interferon, that's a recipe for severe disease of many different viruses. So it's a risk factor for severe COVID. It's a risk factor for severe dengue and West Nile virus and probably others as well. Innate immune system, really, really important. Yeah. So John says, in the 2020 episodes of TWEVO, it's so clear from the phylogenetic standpoint that SARS-CoV-2 came from nature. The evidence hasn't changed except to reinforce zoonosis. Every new paper that comes out reinforces zoonotic origin. No new papers come out to reinforce a lab origin. There's no data. There's just people speculating. Here's the biggest one. They were doing gain of function with a coronavirus, so that could have led to it. Okay, first of all, they were using SARS-1, which is very distant from SARS-CoV-2, so it couldn't have become SARS-CoV-2. And even if they were, where's your evidence that says it did, as opposed to really good evidence that the virus first spilled over from animals at the market, the Huanan seafood market? Yeah, you can make all kinds of speculations. It doesn't mean anything. And to base a congressional report on is really, frankly, embarrassing for this country because the rest of the world doesn't do that crap. So, as I said, Jay Bhattacharya, Guy who thinks the virus came from a lab, forget it. All right, now the other part, people have said, well, Vincent, he was right about the no masking and no shutdown. No, he wasn't right. Early in the pandemic, he made these suggestions without knowing anything about this virus. And it turned out that maybe some of the things he suggested might have been implemented. But at the time, we had zero information. And this group, Great Barrington, they think they all know everything. And they say, oh, we should be doing this and that. No, you have no clue that what you're talking about would be a disaster or not. So, and then, of course, he did a serological survey, which is just the worst example of science ever, right? He recruited people on Facebook, just totally bogus, too small, making conclusions about infections. So I think the person who heads NIH should know science, and I don't think he knows any science. He's a public health economist or something like that. You know, it takes a lot of work to know science. And he's done his own field, but he doesn't know the kind of science you need to run NIH. I mean, Tim Collins, is that his name? The former NIH director. I didn't agree with him all the time, but he knew science. All right. And that's what we need. All right. So Angry Penguin's biggest concern besides RFK is H5N1 adapting. So you can be concerned about it. I think it's fine. We just don't know whether it will happen or not. How quickly an effective vaccine could be made? Well, it can be, I mean, this may have already happened. It can be given a phase 1 already because we know we don't know the exact pandemic virus that would emerge, but we know what's circulating now, and we could use those and do phase 1. And then if there's an outbreak, we could do phase 2. So maybe we could have it done in 6 months. So the phase 1 would be already finished. Certainly less than a year, right? That's my idea. So you say it seems a lot closer. No, I don't see the evidence that it's a lot closer. It's in more animals, which, as I said, could lead to more mutation, but I don't think it's necessarily more closer. Yeah. All right, let's see what else here. Oh, so crumble is everywhere. Okay. Well, I would never wait online for anything like that. But they opened a new pastry shop just below the incubator, and it looks lovely. Right, the window display is awesome. Right, many places don't bother with a window display, but they did here, and they have different examples of what they make. It looks great. I haven't gone in yet, but I would go and sample it and support them. Margot says, why is norovirus so high in Arizona, along with crazy numbers of respiratory infections since November? I don't know. Nobody knows, right? Right. Norovirus is a fecal-orally transmitted virus. There could be many reasons. It could be that population immunity has declined due to less infections in previous years. Nobody knows. Respiratory infections. Well, it is the respiratory infection season, right? And maybe now this year people are getting together more than ever because they're happy to be out of the pandemic. I don't know. Till we see some data, don't know. And we won't have data for a couple of years because that takes a long time to get. COVID Arts is an arts student who loves viruses and learning about them. I find them interesting and somewhat beautiful. They are. I love microbe TV and listening to TWIV and virology lectures. I draw viruses myself. show us some of your viruses. You could send them, and we could showcase them here. You could send them to Vincent at microbe.tv. Love to see them. Nevitz, my prediction, H5 outbreak to start on January 25th. Okay, we'll see. We will see. I don't know. I just don't know. We need to put iron lungs together again. Well, during COVID, many people would have liked to have iron lungs because they're easier on the lungs than forced 16 liters of air a minute, right? With intubation. I'm not sure that would happen. They're pretty damned expensive. I mean, I saw one on eBay used, not even functional. It was $35,000. I don't think we'd go. I don't think we'd go to those levels. You know, in the heyday of polio, there were 30 to 40,000 cases a year of paralytic disease. It's a lot of people. I don't think we're going to get to that point because I think enough people will get vaccinated, right? They realize that it's important to do that. Margot says there's avian flu in wastewater here. Yeah, so it's in the environment, and who knows what the source is, right? It doesn't necessarily mean human. Even though it's human wastewater, there can be other things getting into it at different points of the stream, right? So animals could get in as well, so we just don't know. Hello, H-Sue. Good to see you. Thanks for your support of science communication. Really appreciate it. We need it. Nick Zentner, who does live chats on geology, gets people with questions to him to use capitals so he can see them easily. I don't need capitals. I can see them. I have a little teleprompter right in front of me, and they're there, and I can read that. It's no problem. Okay. Maureen says, Science Channel's Conspiracies Decoded had a good segment on wildlife origins of COVID. Original air date was 1/14/21. Middle segment after Yellowstone supervolcano. It's pretty early on. Very interesting. Kang says, virus is becoming pandemic is like Schrodinger's cat. We can't even open the box. It's right. It's right. Until it starts, right? Hello, Jill in Wilmington, where it's 39F. Welcome. Hungry Ghost says, thanks to this channel, especially Daniel, I'm getting my 4 vaccines this week. COVID, RSV, shingles, and pneumonia. Excellent. Angry Penguin, just an FYI about being ready. Sanofi just built a $950 million facility in Toronto to manufacture flu vaccine. It's quite an investment. Good for you, Sanofi. Is it possible for someone to be both immunodeficient and also have an autoimmune condition? Yeah, because immunodeficiencies don't have to be global, right? They can affect specific parts of the immune response. So you could be innate immunodeficient and have a B cell. And you could make antibodies against yourself. So yeah, both can happen at the same time. Yeah, Baloxavir, that's right. So Daniel doesn't use Baloxavir. He should. The Baloxavir targets an endonuclease in the influenza virus. So that's an enzyme that cuts cellular mRNAs to make primers for the flu polymerase. And last year, the discoverer of the endonuclease, Bob Krug, a virologist who is now retired, but he discovered it way back when. He wrote into Daniel and said, hey, Baloxavir is really good. You just need, you can take it up to 5 or more days after testing positive, and you don't need many doses, and it's really effective. So then he said, I don't use it. I never prescribed it. I don't get that, right? If it's really that good. And Gnevitz tells us that Great Barrington, yeah, it's a very nice town. I've been there. Declaration has nothing to do with it. It was, and I don't think the town was very happy about that because it was a dumb declaration. Yeah. Okay. Family wants to know, in the 60s UK, we weren't given BCG until senior school. Is that when it came out, or is there a reason for waiting? I don't know when BCG came out. Let's look it up. No. It was first used in 1921. So that's not it. I don't really know the answer, but the one thing that comes to mind is once you vaccinate someone with BCG, you remove the usefulness of the tuberculin time test, which is they inject a little bit of antigen intradermally in your arm, and then they see if you have an immune reaction to it, which would tell you if you're infected or not. But you also get a reaction if you're vaccinated. So the vaccine eliminates the usefulness of that test. Maybe that's why they delayed it in the UK. I don't know. Okay, Hans says, Vincent, you once said the next pandemic could be due to influenza H2N2, which caused an outbreak in 1957. From 57 to 68, H2N2 circulated. Okay, why? So actually, Ron Fouchier works on H2N2, and I had him on TWIV a couple of years ago at a meeting in Rotterdam. And he said, basically, anyone born after 1968 does not have any immunity to H2N2 viruses. Yet, the viruses continue to circulate in birds. They went from people to birds. And so they're circulating there, and they could pop back into people, presumably. And that's why he thinks that H2N2 could be the next influenza pandemic, not H5N1. Rach says, have you heard of Stephen Bartlett from the podcast Diary of a CEO? I think it would be helpful for TWIV if you went on his show. He had a guest who was for vaccines, but not the COVID vax. I don't know. These people have to find me. But you say here, Stephen asked for someone to come on his show to give their view. I'd be happy to do it. If it's a legitimate show, I'm happy to do that. Bye, Barb Mac. Thank you for moderating. See you next time. Tom points out that being for vaccines and against COVID is a suspect stance. It casts doubt on for vaccines. I agree. Yeah. Anyway, I'm happy to go on any reasonable show of any kind. I'd like to get Plotkin's book on vaccines, but it's a little pricey. Any other recommendations for books? Yeah. Jeff Puloffa gave me a copy. It's very expensive. So you want a book on like individual vaccines or just general concepts, right? I don't know anything. I have nothing on my shelf here that I can recommend to you. There's a good chapter in Principles of Virology on vaccines, but it's just a concept chapter. It doesn't cover each individual one. That is why the Plotkin book is so good. Maybe you can find it used. Yeah, we're only beginning to better understand long COVID. I hope the GOP will continue to fund it. Well, they're going to be under a lot of pressure, right? Because their constituents will say, hey, we have long COVID in this state. Why aren't you funding this research? If you don't, we're not going to vote for you next time. as it should be, right? Spamless says, Vincent, would you know if you were arguing with an AI? I got to chat with an assistant on Amazon. I asked her if she was an AI. She said she was a human and gave a name. I don't believe her. I think you could tell if it were an AI. I think there would be aspects of the interaction that would be weird, right? Because it's not perfect yet. If you listen to that Google AI thing that you put a scientific paper into it and it makes a podcast with two people chatting about it, it's a little off, right? Same thing with written stuff from ChatGPT. You can tell it's off, right? So I would, if you could talk to them long enough, I guess you could figure it out. That's so weird, right? It's so weird. Yeah, I've heard this. Finland has been offering a bird flu vaccine for persons at risk since June. Yeah, that's very good. Speaking of norovirus, I heard there is a vaccine coming. Why wouldn't the cruise lines be all in? Immunity doesn't last more than 2 to 3 years, I've read. Yes, a vaccine is being worked on. I don't know what stage it's at. We'll certainly be covering it when it's released. I don't know why. We used to have a cruise line person listen to him. I think it was from Norway, and he was responsible for, cleaning up and stuff. He showed me once the new sinks they designed where you put your hands in and it cleans them up and so forth. I don't know what the situation is with the cruise lines. You say, why wouldn't they be all in? I don't know. Maybe they are supporting it to a certain extent. I mean, if I were working on a vaccine, that's certainly where I would go for some financial support, right? All right. So Peter says, has the info from TWIV 1013 changed? Let's see what that is. TWIV 1013. Bird flu around the world. So that's June 2023. Another good one would be, let's see, 1053 bird flu in Baltimore. So we had a couple of bird flu experts on the panel, which I think would be useful. And then a more recent one with Richard Webby. Let's look that up. So 1053. And Webby was 1113. I think they cover all. So the 113 covers the cows, and that's the latest one that we did. But the one in Baltimore is really good also. John wants to know, what are your 2 top memorable pieces of art related to virology microbes that you've seen or had an idea for? My friend is making me a small drawing. Oh my gosh. What's this guy? I saw a piece of virus art in person once at a exhibition in Crystal City, right outside of DC. And let's see if I can find it. The guy's name. Yeah, I can't find it, but it was bigger than life. And this guy repurposed computer parts to make virus art. He made an adenovirus with mainly CDs and other computer parts, and he also made a phage, which is just gorgeous, made of various metal parts. I don't know his name, but I after I visited that exhibition. So his work was there. He wasn't there, but his work was there. I took a lot of pictures and I posted them. So I have to look it up. I don't remember. And he's got a website still, and you can see pictures of his art. It's just beautiful. I mean, I like a lot of virus art, like Michelle Banks does a lot of great watercolors of viruses of all sorts and other things. Like this picture behind me, you can't really see details. It's a watercolor of different viruses by Michelle Banks. But I liked his because it was made of unusual parts, right? Virus are made with computer parts. Let's see if that does it. Oh, yes. Yes. T9 track virus. Yeah, a representation of T4 phage made with PC power supply cables, CD-ROMs, floppy drive parts, and 9-track magnetic tapes. So let me see if I take that and search for it. I'm trying to get you his name so you can go look up his website. Oh, I got it. I'm going to paste this in the show notes. This is the T9 track virus, but he's got some other things in there as well. Okay, let me put it in the live stream here. Here we go. I like those a lot. And of course, Mark Martin, right? Yeah. Spamlet says, I used to drive through Highgate at 3 a.m. There's usually a long line of people queuing for bagels and black rye bread. It's very interesting. I mean, I think if people have a passion for something, they should wait online, but no. How far can phage therapy go? I'm really concerned about antibiotic resistance. That's a good question. I don't know. I mean, I think for topical infections, skin infections, it can go very far. I think for systemic infection, where the bacteria are spreading everywhere, it's going to be harder because I think a problem may be making antibodies against the virus, although, we spoke with some people down under last year on TWIV, and they said antibodies are not an issue. But many people are working on it very hard. I think it has a lot of promise. There have been a number of success stories, also a lot of failures, right? It's not ready to go yet. I mean, part of the problem is you have to tailor the phage to the infection, right? And that's not easy to do. It can be done. You should read Stephanie Strathd's book. What is it called? Perfect Weapon or something like that. Her husband's life was saved by phage therapy, and she now has quite an effort going to make it work. And she talks about that in the book. Yeah. PPD-DO is not done much anymore. We stopped using it recently. Go with Quant Gold. Yeah. I got PPD multiple times as a kid. That's what I'm basing my thoughts on. Hello, Rima. How are you today? Okay, Rad says, The Diary of a CEO is a total quack podcast. Okay, thank you for saying that. That's why I caged my ability to be on it, right? Because I didn't know anything about it. You would benefit from an Intro to Virology textbook. I mean, Principles of Virology is very good, right? It's something like $180, although you can find used versions easily. And you could go for that. And maybe a library too, yeah. If I remember correctly, I feel like I listened to this program or maybe something similar through the Science 360 radio. Yes. So NSF, National Science Foundation, had an app called Science 360, and they collected all the science podcasts and displayed them 24/7 on the app. And of course, you could also, if you went there, it would just pick one to play, or you could pick your own. And they discontinued support, unfortunately. Really too bad, because it brought science to the people. And yes, all of our podcasts were on Science 360 Radio. So those podcasts are made by Microbe TV, which is a production company, and I'm this Livestream. I'm the founder of Microbe TV, and this Livestream is part of it. Yeah. Hungry Ghosts Plotkin book for $80. There you go. Yep. All this book talk reminds me I want to buy The Cutter Incident. Yes, you should buy The Cutter Incident by Paul Offit. Really good. Really, really good. Highly recommended. Yep. Okay. What else we have here? Thank you, Dave, for your support of science communication. We need everyone's support to make this work, to make science accessible to everyone so people can understand what's going on in the next few years. Am I more worried about bird flu this year than I have been in the past? I would say I'm equally worried as I was before. I don't think it being in so many more animals worries me more. It's been around so long and it has not changed to the point where it needs to be, that it kind of makes me think that it's not possible. But I don't know, right? Yeah, they used to do an art exhibit in Crystal City, and one year they had this fellow whose website I just gave you, and his name is McClure. Let me see. Let me go to the home page. Where's his name? Doesn't put his name on it. Contact. Forrest McClure is his name. And he was there, and so was Michelle Banks, actually. What about the big painting of Ebola? Right. I forgot. That a big painting by who did it? David Molesky. That's pretty cool. I like that a lot too. Thank you, Mike, for reminding me. Jill says, it's cool that the artist made a computer virus. Yes. 30computers.com. 30computers.com. His goal was to make 30 different viruses from computer parts. And he's got pictures of a lot of them. Yeah, it's so cool. Vanity says, someone was watching a New Year's Eve show on my flight last night. I could just caught a quick glimpse of their screen showing the ball, and my first thought was icosahedral. The second was New Year's Eve. Yep, got you thinking right. Ball is not too far from the incubator, by the way. Yep. The Plotkin book, by the way, is just called Vaccines. And Plotkin is one of the authors, but it also has Paul Offit and many other people as well. So yes, influenza B has HA and NA, but they don't undergo the subtypes as does A. So you don't have H1, H2, H3, etc., NA, N1, N2, and 3. It's just HA and NA. And the way you differentiate them is by genome sequence. You can tell if it's a flu B or a flu A. Yeah. The glass viruses at the microbe section in Amsterdam Zoo were great. Oh, cool. Send some. I'd like to see. Vincent at microbe.tv. Okay, Jill says also avoid diarrhea of the CEO. Okay. Yeah, we don't get a lot of traction, unfortunately, but we get enough. I mean, look, tonight we have 188 people on New Year's Day. That's damn good. Thanks, folks. John writes, everyone has been so concerned about a new flu. Aren't there other bat coronas that can attach to ACE2? Would one of those potentially rip through? Yeah, there are. But these are very rare events. You have to have the right virus mutating in the bat somewhere. And then that has to get into people. It's not easy, right? Because mostly bats are isolated and there's not a lot of contact. You have an intermediary animal that gets infected from the bat, and then the animal gets brought to market. So yes, we'll probably have another corona pandemic one day. But look, COVID was the first one in modern history anyway, pandemic, SARS-CoV-2. It took a long time. So I think these are very rare. They're very scary, and that's why people don't want them to happen at all. But they're very rare, which is good, so we can prepare. Dave writes, can you bring Stephanie in for an interview? Yes, in fact, so last year I did a South by Southwest where I interviewed Stephanie and a CEO of a small company that was doing phage therapy. And she's very happy to come on TWIV and tell her story. No, it wasn't easy. A lot of people were involved. It's a good story, but I will bring her on this year. Yeah. Yes, Mark Martin had her on Matters Microbial. Yep. So you can hear a short version of that, right? How about appearing on MedCram? Well, Daniel Griffin was on MedCram. I think that's enough. I'm not a doc. I'm not a medical doctor. I'm a scientist. I feel like Scotty, right? I'm an engineer, not a doctor. Paso Robles. Very good red wines. Uptick of pertussis in Michigan. Yes, I've heard about that. Don't know why. A lot of pertussis. In fact, in the latest clinical update, I think there's a nationwide uptick in pertussis. Yeah. Yep. And thank you, SRR, for your contribution to science communication. I think we could do a a mini lecture now. How's that? I just have a 30-minute mini lecture here, which will give us 15 minutes at the end for questions. So we're going to talk about structure of viruses. We haven't done that. And by the way, in 2 weeks, I start my Columbia University Virology course again for the, 14th or 15th year. And all the lectures will be on YouTube again. I try and update them every year to make things current. So you might want to check them out. So, structure of viruses. We have, in terms of structure, really 3 different kinds of viruses. And I tell my students, just remember the number 3 subway. That's the icon. You remember, there are 7 different genome types. You just remember the number 7 subway icon. There are 3 structure types: helical, icosahedral, and complex. And we're going to explain what these are. But first, some definitions. We can talk about subunits. That's a single folded protein chain. So those are the building blocks for the viruses. The structural unit is the unit from which the capsids or the nucleocapsids are built. They're usually 1 or more subunits. So here's poliovirus, where the structural unit is composed of 3 proteins, VP1, 2, and 3. So that's a structural unit with 3 subunits. And then the capsid is the protein shell surrounding the genome. Some viruses have envelopes or viral membranes. like influenza viruses and herpes viruses. These are host cell-derived lipid bilayers. And then in them are, of course, glycoproteins, spikes. And finally, nucleocapsid is what we call the nucleic acid protein assembly when it's a substructure. So here on the upper right is poliovirus. It's a capsid. On the bottom is herpes virus. Now, it basically is a icosahedral capsid surrounded by a membrane, so it's a substructure. So the membrane makes it a substructure, so we call it a nucleocapsid. And then virion is the infectious virus particle. We reserve virion for only those particles that are infectious. And when we talk about viruses, of course, they're really small. And we have different kinds of units that we use, like a nanometer. We talk about nanometers all the time. Polio is 30 nanometers in diameter. Ribosome is 20 nanometers in diameter. So a nanometer is 10 to the minus 9th meters. That equals 10 angstrom, which is 0.001 microns. Now, just to give you some perspective, an alpha helix in a protein is about a nanometer in diameter. DNA is 2 nanometers in diameter. Ribosomes are 20. Polio is 30. And Pandora virus about 1,000 nanometers in length. All right, so we use nanometers a lot. Now, when you build a virus particle, symmetry, for the most part, except for these complex viruses, symmetry is the key. And this was actually a concept started by Watson and Crick. You know, they discovered the structure of DNA, but then they worked on structure of virus particles. They noticed that most virus particles were either spherical, like this down here at the bottom, or rod-shaped. Rod-shaped. And their idea was, as virus genomes are small, now in the 50s and 60s when they were studying this, yeah, all the virus genomes we knew about were small. So they said, well, this makes sense because the genome is small and you can't code a lot in it. So what we'll do, what we think happens is that the structural proteins are encoded in the genome, but just one copy, and then the proteins are repeated in the particle many times to give it symmetry. And that way, a small genome can encode a reasonably sized virus particle. And so they said, for these rod-shaped viruses, these identical protein subunits are distributed with helical symmetry for the rod-shaped viruses. I'll show you what that means in a sec. And icosahedral symmetry for round viruses. Think about the ball dropping over Times Square, as Vanity just said. So there are two symmetry rules for building viruses. So remember, a subunit is the protein that's making up the virus particle. Each subunit has identical bonding conditions with its neighbors. So each of these subunits here bonds to its neighbor identically, but it's non-covalent. All right, it's non-covalent, so it can come apart, and you get a symmetric arrangement. So non-covalent allows you to not only reverse during assembly, but to get the particle open when it gets into a cell to release the genome. And if you have one protein repeated many times, or a couple, they have to interact the same way, and that way it works. So you can use one or a few proteins to build a virus particle. So today we use this idea of symmetry and self-assembly to make vaccines. So many capsid proteins, if you make them, they will assemble into what we call a virus-like particle, which is simply a virus without nucleic acid, VLP. And the HPV vaccines and the HBV vaccines are nothing more than VLPs made in yeast. So here's an example for the human papillomavirus vaccine. We take the gene for the capsid protein, we make it in cell culture, it assembles into pentamers, which is 5 copies of the protein, and then the pentamers assemble into empty particles. So the protein, the amino acid sequence of the protein, has within it all the information to fold into a virus particle. It's really remarkable. So this human papillomavirus vaccine and the Hep B vaccine, similar idea. Very, very cool. Okay, so helical symmetry. Again, we have, in this case, tobacco mosaic virus, the first virus discovered. One protein, one coat protein, which interacts with each other and which the viral RNA over and over. So here is actually it's an electron micrograph of the particle made up of individual coat proteins. And here they're shown schematically on the right in yellow. Each coat protein interacts with the next one in the identical manner, and they interact with the viral RNA, which is shown here in green. And they make these long rods. So basically the coat protein is protecting the RNA, and they make these long rods, which are shown down here. So that was the first virus discovered, tobacco mosaic virus. And it's a great illustration of helical symmetry. You see, you understand where helix comes from. This is simply a coiled structure here made up of multiple of these capsid protein or coat protein subunits. So that's a plant virus, tobacco mosaic virus. There are animal viruses that have helical symmetry. This one is a paramyxovirus Sendai virus, but measles virus would be the same. Again, here we call it the nucleoprotein interact with each other and with the viral RNA to form these long helical structures. And in animal viruses, any of them that have helical symmetry are always enveloped. Don't understand why that is, but there's always an envelope around them. Whereas in plants, there are lots of examples of helical viruses that are non-enveloped. So here you can see an electron micrograph of a paramyxovirus. This one is broken, and the helical nucleocapsid is spilling out. We call it a nucleocapsid because it's a substructure. It's within an envelope. Here, it's a capsid. There's no membrane around it, so there's no substructure involved. Students get totally tripped up on this every year, but it's a very simple concept. If it's a substructure, it's a nucleocapsid. If it's a capsid, it's naked, basically. How do you get a substructure? One way is to put a membrane around this nucleocapsid. Okay, so here are some examples of enveloped RNA viruses with negative-stranded, single-stranded RNA and helical capsids. So we just talked about helical capsids, right? Talked about tobacco mosaic virus. Paramyxoviruses, measles and mumps viruses, they have a nucleocapsid, which is RNA wrapped around, protein wrapped around RNA, enveloped. Rabies virus, same thing. Influenza virus, and the nucleocapsid is an RNA protein complex with a membrane around it. Filoviruses, Ebola viruses, same thing. They have an RNA protein nucleocapsid surrounded by a membrane. So once again, the nucleocapsid is the nucleic acid protein assembly that's packaged within the virus particle. There are also enveloped viruses with helical capsids and plus stranded RNA. And you know this one, SARS-CoV-2, right? It has an RNA protein nucleocapsid surrounded by an envelope. It's a helical nucleocapsid like the ones we've been talking about. And of course, I include this electron micrograph of a coronavirus to show you the halo of spikes embedded in the membrane, which was thought to look like a solar corona, hence the name. And here is from an early paper in 2020 showing electron micrograph of some virus they pulled out of the lung of a patient with COVID. And they looked at these spikes and said, looks like a coronavirus. There are other viruses with helical symmetry. They're both DNA and RNA. You have double-stranded DNA viruses of archaea, right? These are helical symmetry. This is enveloped, so it's a nucleocapsid. We have many viruses that look like tobacco mosaic viruses, which are coat proteins wrapped around the DNA. This one is different because it has a tail fiber. That's another virus of archaea. Single-stranded viruses of bacteria. There are many of these helical single-stranded DNA viruses. And then we have both rod-like plus and minus RNA viruses of plants. Again, a capsid made up of a single protein interacting with the RNA, and also flexuous plus-stranded RNA viruses of plants. These are just same structure, except it's flexible. It's not a rod. Okay, that's helical symmetry. What about round viruses? How do you make a round virus? Well, this is not a good example. Here's a good example. This is my TWIV Christmas tree ornament. It's poliovirus. It's round. How do you make a round capsid from proteins that are not round? They have irregular shapes, in fact. All right, so this was figured out not only by Watson and Crick, but also by Casper and Klug. And the first clue is that all round capsids have a precise number of proteins. Multiples of 60 are really common. The second clue, spherical viruses come in many sizes, but the proteins that make them up are always about 20 to 60,000 Daltons in molecular weight. So Watson and Crick said, these viruses are made with icosahedral symmetry. What's an icosahedron? I'm glad you asked. It's a platonic solid. We've known about icosahedra for years, right? An icosahedron is one of the platonic solids. It's a solid with 20 faces, each an equilateral triangle. 20 of these equilateral triangles. So it's a triangle, same triangle repeated 20 times. And that gives you 5-fold, 3-fold, and 2-fold axes of symmetry, which simply means there are 5 copies of the triangle around the 5-fold axis, 3 around the 3-fold, and 2 around the 2-fold. Very simple. Why an icosahedron? It turns out that by using icosahedral symmetry, you can make a closed shell with the smallest number of identical subunits, 60. You could take 60 protein subunits and make a virus particle. And you can only do that with an icosahedral. And it's stable. It's really stable. Neurovirus is an icosahedral particle. It is very hardy. It's not inactivated by passing through your GI tract, nor by alcohol-based hand sanitizers. So it's a very stable structure. The simplest icosahedral capsid, like poliovirus, well, smaller than polioviruses, actually, parvoviruses, are made up of 60 structural units made up of one protein. So remember, the protein is the subunit, and the structural unit is what is making up the virus particle. And in these simple viruses, the structural unit is made up of only one subunit. So you basically have 60 subunits and 60 structural units making up these virus particles. So here's a diagram of it on the left. There's the structural unit. There's a view of the particle from the top. There's the 5-fold axis. And then at the bottom is a capsid. And in these viruses, the interactions of all molecules with their neighbors is identical. Now, even though icosahedral symmetry is used to build them, and we use icosahedra to illustrate them, they're not actually icosahedral in form. They're spherical. The icosahedral refers to the symmetry that's used to build the virus particles. Okay. All right. Here's a simple icosahedral virus particle, adeno-associated virus 2 or parvovirus. About 25 nanometers in diameter, pretty small, 60 copies of a single capsid protein. So the subunit and the structural unit are the same. And inside is a single-stranded DNA genome. But this is the particle here. It has 5-fold, 3-fold, and 2-fold axes of symmetry, just like any other icosahedral particle. 60 copies of a single capsid protein. That's all you need. So the genome only encodes one capsid protein, but of course, in the cell, the genome is translated to make lots of proteins to give forth to the icosahedral capsid. How do you make larger virus particles? The proteins don't get bigger. You just add more of them to the virus particle. So here's an example of a protein with 180 identical protein subunits as opposed to 60 for the previous one. Now, not every subunit is interacting in the same way. You have now what we call pentamers and hexamers. So in the previous example, there were only pentamers. In other words, here's 1, 2, 3, 4, 5 copies of the capsid protein surrounding the five-fold axis of symmetry. But in these now, you have pentamers. Here's one in green, orange, 1, 2, 3, 4, 5 proteins around it. But you also have hexamers. Look at this purple one with the yellow proteins, 1, 2, 3, 4, 5, 6. So by adding additional proteins to make the particle bigger, you're making hexamers and pentamers. That's just the nature of adding more subunits there. But fundamentally, this now changes the way these proteins interact. We can no longer say all 60 copies interact identically. Now we say they interact with quasi-equivalence. They're similar but not identical. So we get tail-to-tail interactions, we get head-to-head interactions, but you can see if there's a hexamer here and a pentamer here, there are going to be some different bonding environments. So they're not exactly the same anymore. So that was Casper and Klug's contribution to virus structure over Watson and Crick. Quasi-equivalence, when a capsid contains more than 60 subunits, each occupies a quasi-equivalent position, which means that the binding properties in different structural environments are similar but not identical. Again, you have hexamers and pentamers. The interactions have to be different when you have 5 proteins interaction versus 6. And that leads to this concept of the triangulation number, right? The T number, which is very simply stated as the number of subunits comprising the structural unit. Okay? The subunit is the protein chain, and the structural unit makes up the particle. So let's look at a T equals 1 capsid. The subunit and the structural unit are exactly the same. So each structural unit has 1 subunit. So it's a T equals 1 virus. And in fact, you can use the T number to figure out the total number of subunits, protein subunits in a particle, by simply multiply 60 by the T number. And so 60 times 1, 60. So these have 60 subunit proteins. Now, a T equals 3 particle is slightly bigger. As you can see here, we've added more subunits. We have T equals 3, which means that there are 3 subunits comprising the structural unit. And you can see that here. This red triangle, one of the icosahedral triangular faces, has 1, 2, 3 subunits making up that structural unit. And that's a T equals 3 virus. So if you multiply 3 times 60, you get 180 total subunits making up that particle. And these can get quite big. As you can see here, we have T equals 4, T equals 13, and that's how we make bigger and bigger viruses. And remember, anything above T equals 1 has not only a 5-fold, but a 6-fold axis of symmetry. Okay, so a number of years ago, I learned how to make icosahedral viruses with these tiny magnets, incredibly strong tiny magnets, which you can buy at Amazon. I have hundreds of them. And I made a video showing that. So it's really very short and very much fun. But here's a T equals 1 particle, right? 60 subunits made of all pentamers, which means 5 subunits making up the structural unit. Sorry, these are the 5 proteins that are repeated 12 times to make up the virus particles. You can see there's 1 pentamer there, there's a second pentamer there, and so forth. On the right is a T equals 3 particle. This has 180 subunits. And now, since we've added more virus subunits, or more than T equals 1, we have pentamers in red and hexamers in blue. And you can see how they are interspersed in the virus capsid. Here's a pentamer, 1, 2, 3, 4, 5 in red. Here is a hexamer, 1, 2, 3, 4, 5, 6 blue. So, and here's another hexamer next to it and so forth. So you could just keep adding more subunits and make bigger and bigger viruses. I actually have enough magnets to make the next one up, which is T equals 4. I'd love to do that. I just never got around to doing it. Here's poliovirus. Is an example of a T equals 3. It's actually called pseudo T equals 3 because the 3 proteins that make up the 3 subunits that make up the structural unit are 3 different proteins. Usually it's the same protein repeated 3 times to give a T equals 3. For polio, there are 3 different proteins, VP 1, 2, and 3. So we call that pseudo T equals 3. So here it is. We have the structural unit is a protomer of VP1, 2, and 3. So VP1, 2, and 3 are the subunits, the protein subunits, and those are repeated 60 times to give you the intact particle. And so here's a model of the particle. You can see our 5-fold axis of symmetry right there in blue, and here's a 6-fold axis of symmetry. This is bigger than T equals 1, so it has hexamers as well. Okay. Of course, we can make even bigger capsids like adenoviruses by simply adding more subunits. But there are also specialized proteins in these larger viruses, like these fibers in adenovirus that are used for attachment to cell receptors. As viruses get bigger, it's very difficult for them to be stable with hexamers and pentamers, so there's some other proteins shoved in the capsid, which are called cement, to help them be more stable. And there are proteins that interact with the DNA and so forth. Now, capsids can be covered by host membranes. These are envelope virus particles. The envelope is derived from the host cell because viruses can't make lipids. And the envelope is acquired by budding of the nucleic capsid through a cell membrane. These nucleic capsids can have helical symmetry or icosahedral symmetry. So here's HIV budding from the cell. It has an icosahedral symmetry nucleocapsid. When you have an envelope derived from the host cell, then the particle must have spikes, integral membrane proteins that are embedded in the envelope and allow the virus particles to attach to receptors on the host cell, for example. And these glycoproteins, for example, the influenza virus HA, this one is perpendicular to the virus membrane. The flavivirus glycoprotein is actually lying parallel to the membrane. Either works and allows for virus entry. So here are 2 kinds of envelope viruses. On the left, we have an envelope virus with a helical nucleocapsid, right? The influenza virus, Ebola virus. On the right, we have envelope viruses with icosahedral nucleocapsid, herpes viruses, and Toga virus, rubella virus. And finally, we have this class of viruses which are very big, the pox viruses, the Pandora viruses, the pithoviruses. They're 1.5 microns in length in some cases. You can see them in a microscope. They have no repetitive properties as far as we can tell. There's no symmetry. Rather, they're made up of a variety of components, membranes, lots of lots of virus proteins. And so we call them complex virus particles because we don't understand how they are built. Okay, so that's a really brief primer to virus structure to start you off in 2025. Let's see if there are any questions here. What was the last one that I answered? Okay, Paso Robles. Okay, so Lisa says Stephanie Stratti gave a lecture at OSU, and it's archived at Ohio State Science Sunday. That's great. So you can check her out. Gary, thank you for your support of science communication. Really appreciate it. Absalom wants to know about prions. And Absalom, we did a prion mini lecture last year, I believe. Prions are fascinating. And there is one also in the virology course. So you should go to those two resources to check it out. Yeah. Yeah. Tryon says, does intradermal inoculation of antigen induce secretory IgA? I don't believe it does. I believe to get secretory IgA, you need to be at a mucosal surface. Okay. I could be wrong, but that's the way I see it. Nevitz Forever says, I worked on the development of the original HPV vaccine, quadrivalent. Hundreds of people and years of work to get the proteins to behave, not aggregate, retain potency, etc. Remarkable. Public has no idea. No, the public thinks that it's just big pharma wanting to make a lot of money. They don't understand that they do want to prevent cancer, and they have to charge for it, right? That's what capitalism is all about. But yeah, a lot of people put a lot of time into these, and a lot of people dismiss it. It's just unfortunate. Yeah, G worries about prions in deer. Yeah, deer and elk have prions, and they're spreading across the U.S. and other parts of the world, Canada. And we're worried that they may enter the food chain, the human food chain, and cause diseases, right, neurological diseases like mad cow disease and bovine spongiform encephalopathy, and the human version in the UK from people eating beef from cows that had a prion disease. So we'll see that. Thank you, Rach, for sending those pictures. I'll check them out tomorrow. Yep. How are subunit vaccines different from a capsid? So a subunit vaccine, you take like the spike of SARS-CoV-2, and it's one protein. It trimerizes, but that's it. A capsid vaccine makes the shell, the icosahedral shell. It's empty, but it looks, for all purposes, like a virus capsid. If you took a picture of it, it looked like a capsid. Whereas when you make the subunit, whether it's the glycoprotein of shingles, varicella zoster virus, or SARS-CoV-2, it's just a protein subunit, and the best it does is trimerize. Yeah. John writes, when I see the icosahedral shape, it makes me think I understand. But then when I see the stringy zoom in of what it looks like, I get a little overwhelmed. Right. So you have to imagine that they're not stringy, but they're full of atoms that are taking up space. So the stringiness is one way to depict. But let me go back to another way we did it for polio, which is better, I think. Let's see here. Yeah. So let's play that. So here on the right is poliovirus. Now we've depicted it where every atom is a sphere. So now it has substance. It's not this stringy stuff, right? The stringy stuff is just actually what's called the trace of the amino acid chain. It eliminates all the side chains. And here we've got all the atoms, and it can inform you about how they occupy space, right? So I think that helps a lot. Okay. Oops. We hid eCam. Okay. What else do we have here? Imagine it took nature millions of years to figure all this out, so don't feel badly if you don't understand it. Just enjoy the awesomeness. The host cells would contribute to the number expressed. So yes, the translation, the protein synthesis is occurring in the host cell, right? And so the rate of protein synthesis will determine how many virus particles can be made. And some cells may not make a lot of viruses because they can't make enough capsid protein to make lots of capsids, and others can do a good job. But yeah, it's up to the host cell to make enough. Sometimes viruses tweak the translational apparatus to make it go faster, but it is the host cell that's making the proteins, yeah. So Jill writes, the tiny mag buckyballs are now naturally illegal to sell because kids were eating them. Yes. So for a while you couldn't get them on Amazon, but they're back. You can go and get them. So I don't know what's going on, but yeah, the kids were eating them, and that was not good. Not good at all. Question: in virus assembly, can RNA DNA cargo request envelope particles to cover me, or envelope parts say hide in us? Who is initiating the assembly process? Yeah, it's a very nicely phrased question. Phrased questions. I like that. So, the assembly process of the icosahedral capsids initiated by the proteins themselves. The way the nucleic acid gets in differs depending on the virus. Sometimes, like for herpes viruses, there's a hole. There's a single hole called a portal. I don't know if anyone remembers a video game called Portal. Oh my God, that was so good. There's a portal through which the DNA enters the capsid. So once the capsid is formed, it gets in. Sometimes the RNA, as for poliovirus, binds a pentamer, which is just 5 copies of the structural unit, and then that assembles into a virus particle, pulling the RNA with it. Now, for envelope viruses, like some retroviruses, they form from the cell surface by budding. The membrane forms, but the components are brought there by a cellular process that drives this budding. So it's a mix, really. Sometimes it's cover me or hide in us. Very nice. Why is Pandora virus not a box? You know, Pandora's box was actually a flask. It wasn't a box, but the name is stuck. If you see some old pictures, drawings from antiquity, you'll see she's holding a flask, which is more like the virus, I think, than a box. But the reason they called it Pandora virus is because when the genome was sequenced, they found lots of genes encoding proteins. They didn't have any idea what they did. So mystery. So you open up these viruses. It's like opening Pandora's flask, right? That's the idea. Good question. Why doesn't Purell work against non-envelope viruses? So norovirus is not enveloped. Neither is poliovirus, because those icosahedral shells, the proteins are so tightly bonded to protect the nucleic acid. It's very hard. The only way they come apart is by attaching to the right receptor. So, yeah, ethanol is not going to work. Ethanol mainly works for envelope viruses. It dissolves the membrane around the virus. But for noroviruses, nope, not going to do it. In Kang acid, in budding viruses, are the viral proteins lined up near the budding site? Yes. So the glycoproteins line up. If the budding is from the plasma membrane, not all viruses bud from there. Yes, the glycoproteins line up. Some bud into internal compartments, but again, it's the same thing. The glycoproteins all line up in patches on the cell membrane. Yeah. Dave wants to know if the size and number of subunits offer any indication or measure of complexity of antigen and vaccine development. So for icosahedral viruses, you typically want to make the capsid, right? Usually an empty capsid. So, you know, in that case, the size doesn't really matter. But for envelope viruses that have glycoproteins, the larger the virus, the more glycoproteins are found in the envelope. So, you know, SARS-CoV-2 has a couple, and the spike is the most important, apparently. But herpes viruses can have many, 8, 9 glycoproteins. So you have to figure out which one you need to use to get protective immunity. So that does complicate vaccine development. It's a perfectly good question, Dave. Absolutely. Absolutely. Vincent, myself, and a few others and a Canadian virologist are going to some high schools in February and March for some virology presentations. What should we teach the youth? I think you ought to teach them. Well, you're probably only going to have an hour, right? I think you, I don't know how much youth cares about the origins of SARS-CoV-2. I mean, it's a fascinating story, right, to recount it as it happened, but I'm not sure that they're interested or they care. But if you think they're going to go home and tell their parents and change their minds, maybe that's a good way to do it. I like showing structures of viruses because they're all, you can have beautiful pictures and distill them into concepts like I talked about today. I think that's fascinating. And also I think it's important to emphasize the extent of the virus sphere. There aren't just a few viruses out there. They infect everything. There are billions and billions of them. And they're evolving constantly. I think that is a really important thing. So where new viruses come from, this is really important. Yeah, Will, I remember you loved the first lecture last year. Yeah, the first one is very inspiring for me to get them interested in the course right on, right? Yeah, it's really important. What's my favorite virus? My favorite virus has always been poliovirus because I spent many years working on it. And the conundrum we are currently in with respect to the attempt to eradicate is fascinating. And it's going to continue to cause problems in 2025. Will asked me to itemize the topics I'll be covering in lecture 1. Well, it's, yeah, like the nature of viruses. What is a virus? Where did they come from? And how do we study them? With examples. And, it's been very much the same lecture for many years. I may try to tweak it a little bit. But yes, viruses are not bad, always bad. Some of them are, but many of them contribute to us. The origins of viruses and their diversity and how we study that and how they contribute to the diversity of life on Earth. Yeah, for noros and polio, soap and water work. So basically, you wet your hands, lather them up, and rub for, recite the alphabet. And what you are doing is the soap is pulling the particles off of your skin, and then you're rinsing. At the end, you will rinse them off. So the vigorous rubbing is you're decreasing the number of particles attached to your skin, and then you're going to rinse them away. So you're not inactivating them, but you're getting rid of them. This is funny, Nevitz. SDS does not inactivate polio or norovirus. Yeah, 8 molar urea will, but you don't want to use that on your hands. How does the shingles vaccine work given that the latent virus is already in the body? Right. So what happens is you get shingles vaccine and you make a good immune response to the spike. And then when you have a reactivation, you limit the amount of virus produced and you limit the amount of nerve damage and the rash because those antibodies are blocking virus replication, right? It's not zero, but it's enough to reduce the symptoms. That's how it works. How many years do I plan on teaching? I haven't thought about it. As long as I can. I really like it every year. Every year is a joy. I love instilling in students a love of viruses. So I will do it as long as I can. When I can't think properly or even get around. I have to take the subway from the incubator to Columbia. But I don't know. That's my goal, to do it as long as I can. Okay. I think we'll wrap up this first office hours of 2025. I'm going to try and do it every week, travel or teaching notwithstanding. But I want to thank our moderators tonight for helping out. Really appreciate that. We had Tom, we had Les, we had Steph SF, we had Barb Mac UK, and we had Les, or I already mentioned Les, right? I mentioned Tom, Les, Barb Mac, Vanity Nutrition. There we go. Thank you so much for being here. Really appreciate it. On New Year's Day, we have 185 people here. Hit the like button, by the way, folks. And to infinity and beyond. Yes, go. Love it. Thanks for coming, folks. I really appreciate it. We'll be back next week. When is next week? That's Wednesday, the 8th of January, 8 p.m. Try to do this every week. And in the meantime, have a happy new year. I hope all of you have a great 2025. There are always going to be problems you're going to have to solve. Don't let it get you down. Just use your mind and solve it, and you'll be fine. Go to look for where the answers are. Use your curiosity and passion that I've tried to instill in you in these past years. Okay, folks, I hope to see you next week. Meanwhile, be safe. Happy New Year.